Hello, my name is Madison Mond, and this is my second local expert project, and I have decided to do it on organoid cultures. So what are organoids? Organoids are tissue engineered in vitro models that are able to encapsulate an organ structure and then the cell type diversity that are housed within that organ, and it's able to represent a number of different types of organs. So the different organs include small intestine, colon, stomach, esophagus, tongue, liver, lung, pancreas, heart, ear, and skin. And so what these are basically mini organs that are able to be grown up and then used for different research models. So the different cells that are able to make up the organoids can be derived from two different types of cells. So there is induced pluripotent stem cells, and then there's also tissue-derived cells. So this is I thought, a really interesting picture that shows how these organoids are actually cultured. So it starts off here with the tissue-derived cells. This is the first version. And so in this example, they are taking cells directly from the gut and the stomach. And what they do is they cut it open and they cut these samples into really small pieces, and then they plate them. That way they can be enzymatically digested or mechanical disassociation, as the goal is to isolate single intestinal stem cells here. And once these are isolated, they go through several rounds of washing and purification. That way they are in their purest form when they move to the next step, which is the seedling and expansion, which then is able to preserve these organoids and then allow them to expand and multiply. The next step of this is the different type of stem cells. So this is the pluripotent stem cell. And basically the goal is to generate these organoids from these pluripotent stem cells using genetic engineering. And what happens is these different stem cells are maintained and then expanded either as undifferentiated populations on different feeder cells or in a extracellular matrix substrate. But the goal is to make sure they grow and they populate and aggregate into these embroid bodies. And so then these are normally harvested as cell aggregates, which preserve cell-to-cell contact and then create higher viability within these cells. So once these aggregates are formed into these embroid bodies, they are able to then form specific germ layers and then eventually go into the seeding and expansion, like the last step, that they can be preserved and then specialized into their organoid of choosing. So my next part is I just want to show you a few pictures of the actual organoids. So this one here is a human liver organoid. And what happens was this organoid was derived from a biopsy from a healthy liver donor. And then this is showing how it expanded in a culture and how there's a budding process so it's able to replicate. This is my second image, and I thought it was really cool. This is mouse intestinal organoids, and these were actually once growing as cancer, but now they are able to undergo a certain type of differentiation and are actively dividing stem cells now. So this technique obviously ties into our cell culturing model. So this is a way not only to grow up the cell cultures, but it is a way to model whole organisms, which then can be used to study a variety of different things. So some things that have been done is diagnostic work, looking at diseases and how they affect the cells within our organs, personalized medicine, and then new drug discovery, potentially new drug targets, all these things. This technique is super interesting. However, there are some pretty big limitations of this technique. So this is a very new technique, and since we are dealing with stem cells here, there is an ethics debate. And the main ethics debate, and the one I found the most interesting, was regarding the use of brain organoids. And the fear is that they will become so specialized that they will be able to become self-aware and the brain organoids will actually have the ability to feel pain. Now, this hasn't happened yet, but this is a possibility in the future that we have to be aware of. And so that ethical side is definitely a limitation. Not to mention, growing up these organoids is extremely expensive, and so work is limited. And the last limitation that I found was actually found within a paper that was looking at intestinal organoids. And so what they wanted to do was they wanted to cold culture their intestinal organoids with microbes. And what they saw was that this may not show an accurate picture of the effects that these microbes will have in a systemic point of view. So in this sense, our intestine has all of this natural flora. And with these organoids, they don't have that. And so it is sometimes hard and can be a limitation to actually see the interaction between the human model, where we have all of these different microbes and good bacteria, and these organoids do not. But we can see the effects on the organ itself. So the primary comparable method to this technique is the 3D cell culture. And so I found this good little diagram that compares the two. And so what this is organoids are mini organs where the primary cells are spheroid-like. Another advantage with the organoid is that it can be cryopreserved, where the 3D cells may have a challenging resuscitation if they are cryopreserved. Another one that's pretty important is that the 3D cells, they have the ability to predict the in vivo and clinical drug response, but organoids have a better prediction of the in vivo and clinical drug response. And lastly, the 3D cells are good for limited tissue for smaller scale assays, where the organoids have that advantage. They are scalable for a larger scale screening. The controls of this technique greatly vary, just depending on the organ that you want to culture and the cells that are included in that. And so that's going to change the procedure. So I never really found any controls within the literature, but the one main constant that I found, and in a way it's a control, is that the use of an extracellular matrix that will support your cells and create the environment that the stem cells are used to is really important. If you use the wrong extracellular matrix to kind of grow your cells in, they're not going to be able to proliferate. And so the different types of extracellular matrices changed depending on what kind of cells you want to grow and what organ you want to grow. But that was the main type of control that was used. This is my first figure, and it comes from a paper that talks about cerebral or brain organoids and seeing if they were able to model human brain development. And so what they did was they looked at different markers over here that signal forebrain markers or hindbrain markers. And they specifically looked at it 12 days, 16 days, and 20 days after differentiation. And what they wanted to see is if they were present in these different parts of their cerebral organoid, and what they found was these were able to grow the markers that a normal human brain development would have. This second figure comes from a paper talking about the establishment and culture of human intestinal organoids derived from adult stem cells. So this basically figure is showing the establishment of human colon organoid from a fresh biopsy of normal tissue. So this is day 0, and this is shown right after the establishment of the culture taken. This is right before the first split, so that right before the proliferation of these cells. And then this picture over here is after the first split, which you can see shows the numerous amounts of cells and how this will form the organoid. Lastly, this figure is from a paper called The Optimized Culture Conditions for Improved Growth and Functional Differentiation of Mouse and Human Colon Organoids. And so I really want to direct your attention to this A section of this figure right here. And so what this is looking at is the different medias that these collonoids, so the colon organoids, are able to survive. And as you can see in these rows especially, there was proliferation of the cells, where in other medias there was no, we started with the cells and they decreased. So this is looking at the optimized media to grow specifically these colon organoids. And finally, these are my references if you want to learn more. Thank you for listening.